So maybe I am the most fresh candidate in this block. I mean, I just joined this week in Gitam University, and I was a postdoctoral fellow in Emory University from last 4.5 years. Before that, I did my first postdoctoral training in National University of Singapore, and before that was my PhD training, which was done in PGIME at Chandigarh, and followed by a couple of years of not couple of years, like 1.5 years of senior demonstrator position in the same lab. So overall, like around 6 to 6.5 years of postdoctoral, around 7 years of postdoctoral experiences, and before that, almost 5 years, 5.5 years of PhD. Before that, I got some couple of years of industry exposure in one of the cord blood banks in Bangalore. So, with that brief introduction, I would just move into my presentation, which is entitled as prostaglandin EP2 receptor, a promising therapeutic target against neuroinflammation. So just a caution that this all work are basically from my postdoctoral training at Emory University. As you understand, I'm just a fresh recruit in Gitam University, yet to find a lab space. I just got a desk to sit. So, almost everything I'm going to present here will be from my postdoctoral work. So coming to a brief introduction for EP2 antagonist, and EP2 is basically a prostaglandin receptor. And we all know that prostaglandin plays an important role in immune response and modulation, and also have some critical role in inflammatory processes in our different organs, like in the kidney, in the liver, and also in the brain. So PG2 comes, downstream of COX2 mediated signaling, where it's like in the EP2 and the PKA signalings take place. And this has been found to be implicated in different neuroinflammatory conditions in brain. And in our lab in Emory only, another group, they have extensively worked on epileptic model, and they have shown that how EP2 plays a very critical role in influencing the inflammation in the epileptic brain, and then followed by EP2 antagonism, how this could be useful for as a treatment strategy. So I used the parallelly same treatment strategy for Alzheimer's, because it was shown earlier that by deleting EP2, it can reduce the amyloid burden in the Alzheimer's brain, and that's where we aim to determine whether an EP2 antagonist, global antagonist, could reduce AD-associated neuropathology. So before that, COX2 also could be one other target, but because of some cardiac side effects, this could not be used further as a treatment strategy, and that's why people have started looking further downstream. And EP2 was found to be one of the challenging targets, one of the beneficial targets. And if you look into this cartoon here, that the PGE2 synthesis, this PGE2 has got these 4 different receptors. From there, EP2 are there, but rather than that, there are so many other syntheses also there, and altogether there are 10 different prostaglandin receptors, and there are parallel bindings also taking place. So we selectively chose EP2 and then further look beyond. Now, when we talk about the in vitro synthesis of a small molecule, like let's say that PGE2 inhibitor, it comes all the way starting at the basic structural chemistry level. This carbon rings have always made me fear throughout my masters, and if you ask me too much, I'm not going to answer you much about those, because those are the medicinal chemists who were there in the group. They mostly worked very hard to generate so many compounds there in the lab, and then from there I have taken it forward. So that's what, like, stepwise, when you come to a drug discovery program, that's where I work there in Emory. You start with generating or synthesizing a molecule. From there, you test its potency, selectivity, solubility, and then look into the brain permeability and the other safety concerns, into the pharmacokinetics, and then further you move into the in vivo analysis. So when I talk about this particular compound, the journey of TG1177, which is named after my PI, Tota Ganesh, that stands for TG11, stands for probably their notebook number 11. 77 is their page number, and then further like in which molecule, in which particular form of the salt they have used. So that's how it is nomenclatured as Tg1177 HCl. So before that, I would just summarize that there were mostly 500 new molecules were screened through high throughput screening, and from there we found at least 4 to 5 different targets, and from there further we carried it forward, this Tg1177 work. Now, once I go into the lead optimization study, that's what I was talking about, that there are so many different compounds related to EP2 antagonists were synthesized, and also a couple of them were commercially available before, but none of them were tested in the brain system, in the central nervous system. And we started using that, and first it was tested in epileptic model and followed by Alzheimer's, which was precisely my project in Emory. And if you look at molecules, they look pretty much jargons to me, but all I know is like if you shift the bonds from a single bond to double bond, or if you shift a nitrogen or hydrogen from one place to another place, basically you start this story all over again. You start from its potency, selectivity, solubility, and then brain permeability, everything, all the dimension, all the dynamics of a compound changes. And eventually by screen and trial and error method, finally we ended up using this particular compound, which is the HCl salt of Tg1177, which is a very potent EP2 antagonist. In the competitive antagonist study, what we did is we used a very specific cell lines to test like how competitively this particular compound can antagonize EP2 signaling. And if you see in this particular curve, this is the Tg1177, that how dose dependently the curves are being shifted to the right side of your graph with the increasing concentration, and that is a good indication that your drug is, a molecule is actually effective in inhibiting. The EC50 value is basically the 50% inhibition. And then this is the Tg1177, and further we got Tg1177 HCl because the 77 was not very good soluble in water. And that's when we found the HCl salt is more soluble, because you don't want a solvent to be introduced into a drug which probably in a later case may not be approved by NIH when it is coming in the human or animal consumption or IP injection, because you need a NIH approved solvent so that they don't create any harmful reaction in your body. Otherwise, most of these organic compounds are dissolved in DMSO, but unfortunately, you cannot use that as a solvent. But you have to play around with that to find out. And this particular graph shows that it is not selective towards other receptor, like the DP1 and EP2 is very parallel, similar to their structure. And if you see that with the increasing dose, this graph, I'm sorry, somebody is, can you please mute?
Oh, sorry.
Yeah, no problem. So if you see that like with increasing concentration, these curves are not being shifted to the right. So that means like they are really not selective to DP1, but selective to EP2. So that says that there's a very good selectivity. Now, this is the anti-inflammatory property of this particular drug tested in a BB2 cell line, and we selectively, pruned these cells with LPS and ONO. ONO is a known EP2 agonist, so that like we enhance or increase the EP2 signaling in these particular cells, and then followed by the treatment of this Tg1177 at 2 different concentration at 0.3 micromolar and 1 micromolar, and you would appreciate to see that with increasing concentration, different anti-inflammatory modulators like COX2, IL-1 beta, TNF alpha, IL-16, INOS, and also mouse EP2 basically is in the mouse microglial cell lines, and you see that those are dose dependently decreasing or increasing, whatever way it is like reacting to that particular modulators. So that says that it has got a very good anti-inflammatory property as far as the in vitro cell culture is concerned. And then probably we would like to move further on the animals. So when you start with animals, you basically look into their pharmacokinetic dynamics, like you inject or give some oral concentration of the drug, and then finally you look into the plasma concentration. And there is a desirable plasma concentration in these mice who were tested from day 1 to day 6, which is above their potency level, which is the KV value is almost 10 micromolar, sorry, 10 nanomolar, and they are further beyond, like almost 100 or 600 or 400, and that's pretty much higher than the expected range. These are basically the same test at 3 different time points in between males and females, the blue being males and the red being females in plasma. And here you see the plasma to brain ratio, which is like at different time points of the sacrificing the animals, we found like blind brain to plasma ratio in between 0.4 to 4, which is once again, a very, very decent range of the drug to be effective in central nervous system. This is to show that this is 300-fold selective to EP2, because the shield KV of EP2 is 300, sorry, almost 10 nanomolar, I said, and the rest of, for the rest of them, they're almost, above 100. So almost 300-fold selective for EP2 compared to the other prostaglandin receptors. Now moving to the animal studies, I used a C57 5x FAD mouse model of Alzheimer's disease, where you have all the 5 familiar mutations, and we basically given a second hit of this to this mice using like injecting LPS, so that there will be a second layer of inflammation, because in AD mice we initially tested there was not much amount of the LPS, but also we wanted to prove this hypothesis, what happens to the second layer of infection if these mice or the humans with AD, they're subjected to a second layer of infection like viral infection, or let's say COVID in fact. And then finally, how these drugs would be reacting to that. So we tested those mice, and then we looked into the brain inflammation and the amyloid pathology. Interestingly, with LPS, we saw that most of the blood parameters have shown a anemia-like inflammation in the blood, like you see the RBCs and lymphocytes are going down, whereas the monocytes, neutrophils, and both of them going up. And you see the cell density of RDCs and PWGs platelets are also going up. So basically, they are being enlarged. And the hematocrit and hemoglobin, they have gone down. So all these signals are basically putting or pushing the cells, blood cells, towards anemia-like condition. Interestingly, this drug didn't show up any result on the any positive outcome on the bloods. I didn't show it because of the time constraint, but they have shown some good amount of inhibition in different neuroinflammatory markers in the brain, and that too, especially in the female mice, we saw that. That probably one of the reasons, because in the female we got higher amount of inflammation rather than the males, and we saw that you see in average most of these most of these cytokines and modulators have been reduced. Even the microglial and astroglial markers also have been cut down by this drug treatment. They were tested from 2 months to all the way till their 5 months of their age by oral delivery. And interestingly, this was further supported by the protein expression levels of GFAP and IBA1 at different brain levels, which was identified by immunohistochemistry, and also further about the counting analysis at different brain regions.
Sorry, for time constraint, may I request you to conclude and talk about some challenges also?
Sure, I'm on my conclusion slide. Thank you. So overall, this study showed that this particular drug, EP2 antagonist, has got a very good potency and also very selective towards EP2 receptor. And further in the in vitro study, in the in vivo study, we could show that in a 2-hit model, it could attenuate pro-inflammatory mediator, but selectively. And it could be due to the microglial and astroglial inactivation, what we have seen. So these are the couple of papers what we got from our work. One was from the medicinal chemistry background, and another is the Journal of Neuroinflammation, which recently we published. And these are my lab members. I acknowledge my PI, which were Dr. Tuta Ganesh and Dr. Ray Dingledine. And Dr. Ray is not present in this particular photograph, but the rest of the members are there in that. And it has been a very productive, almost 5 years in Emory. And this was basically funded by a couple of NIA grant, which was given to my PI, Dr. Ganesh. So, now when I come back and, talk about the opportunities and challenges, I will keep it very brief, and maybe I will leave it to the audience who have very, very specific questions. Because, if you have already talked to many PIs before, or you have interacted in different Sci-ROI platforms, I'm sure many a times you will be, listening to repetitive answers, like what we should do, what we should not do. So I'll not really go into much details of that. And even I am also not qualified to answer also, because I just moved there. So maybe I can mostly answer your questions related to the application process and how to, go for those interviews and all those things. But when I say so, I precisely put these two pictures, which I have collected, like I have taken back in December 2020, and maybe within a span of 2 days, I have collected these two images. Like one is the video in the right side, and one this, and both of them are on the way to my lab in Atlanta. And this couple of images, what I put, because they actually, gives you the overview of the opportunities and challenges both together. And you see, like, while the red one at the left side, you see there is a beautiful path ahead, and it is basically welcoming you, but you'll have to understand that to get that, lots of flowers from like the leaves, they might have fallen down from that, and then eventually you got that. So what I want to interpret out of this is, you have to basically convert those changes into the opportunities. And the right one, once again, like the same time during the spring season, you see like only a couple of them, or 3 or 4 flowers, or maybe some leaves which are at the top, probably is giving you the indication that which way is wind is blowing. So that tree also must have gone through so much of stress during that period. But all I need to say is networking is one of those, and I know there are some avenues, like let's say Sci-ROI is such a platform. I have been associated with it for the last couple of years, and it has been very, very productive, I would say, like in terms of communicating, in terms of collaborating, in terms of preparing for your next job interview or preparing for the job search. So try to look for such avenues and be connected to that. I know many of us may not be as vocal as some, but just passively be there and try to get benefited. And if you think that you can deliver, just do it. And when you come back to India, the one thing I would say is, like, you basically shed off that baggage of expectations when you are working in outside India labs. The dynamics are pretty much different here. Otherwise, you will be very easily frustrated. But one thing for sure, this country is trying to improve, and that sense is palpable everywhere. So I'm sure things will work out, whoever is willing to come back. And I hope, I can talk in the same tone, maybe 5 years down the line, when I try to establish myself in one of the institutes in India. So with this, I would just like to thank you. And this is basically the science building where I sit now. And if you have any questions, you can always write back to me. Thank you.
Thank you, Dr. Abhijit. It was great to see your talk. So I see there's one question. So one question. Sorry, my son is giving me a hard time. So yeah, so Dr. Aman is asking from JNCSR that what is the half-life of TG11-77?
Yeah, it was there. The half-life basically it's half-life, let's say 2.4 hours, and that is pretty much a valid window, because we don't want the plasma half-life too high, because otherwise they will be denatured after being in the blood circulation only, and we want them to be expressed in the brain. So 2.4 hours, that was a pretty decent window.
Great. Then I may ask the next question. So now you have come back to India, so you must have thought of like making research proposals and like applying for grants. So how did you think of it when like you were moving from US to here? You were like a researcher there under somebody's. Now you have to think of your own research. So did you get like enough idea during PhD or postdoc, or how did you formulate it? And how are you going to translate it here?
Right. I mean, that's pretty challenging, and many times it deserves much of your time and attention. But I would say that if I am willing to come back to India, I would start thinking about it or initiate that process at least 2 years in advance, because you have to train your mind that what you are going to go at. And Dr. Piyush, you would also agree, because you are already part of one of those institutes in India, that you have to know little bit more about that particular institute also before you go there, that what are the facilities are waiting for you. You can't just think of something which cannot be delivered. And if that facility or something is not there, you just out of the way you propose something and you are not able to deliver that, that will be very much challenging. And what I heard after talking to some of the PIs here in Getam, those who have received the seed grants or the preliminary grants, they say that the funding agencies are pretty much flexible towards your first seed grants or like, let's say, transition grants. But after that, they will be very critical. So when you are proposing that, make sure that you can deliver that. Like, let's say at Getam, I don't have an animal facility. I propose that I am going to push, or maybe some of the other faculties, if they are willing, will come forward and push the animal facility. It is there, but it is not in a good shape to perform animal experiments. So we'll have to move that part into the later stage of the project. So let's say if I have applied for a 5 years grant, make sure that those things are in place. So right now, probably I can optimally start with some cell culture works there. And I know there are some cell culture facilities there who are willing to help. I can do there. But establishing a new lab also is pretty much challenging. If you are not into a very hardcore research institute, it is further challenging, because convincing the management also is a little difficult, because they would really not have that research mindsets. To convince that look, for each an individual PI, we need a separate lab space. They might think, no, you don't have a fund. Why do you need a space initially? Just go and sit with somebody else, try to prove yourself, try to get money, and then probably will consider. So those challenges are very much there, but it's doable. I am at that beginning stage. I just applied for one grant and probably will be applying for one or two in the next couple of months, and we'll see from there how it goes.
It's definitely very challenging, but I'm sure you will do great. And so one last question. So did you move with the whole family, and how are they responding to it? So are you getting all support from everywhere?
Yes, I mean, I moved with my family, and that's what I told, right? The process starts very much at the earlier stage. Like, my son is 8 years old, and probably 2 years before I started, that conversation at home that, look, we are not going to stay here forever in US. We have to move. Although I was not sure whether I'll be moving or not, but at least that conversation already started at home. My wife was working. I feel bad about it, because of me, she had to do that sacrifice. She came, but she never complained about it. I hope she gets something here as well to work. But we were pretty clear from the beginning that we came here for the training and we want to go back, and that's how the mindset is. So we, involved ourselves in such a way from the very beginning that we should not be laid back by the American system, that no, things are pretty much better here, and that would rather be forceful on me to not to come back to India. So yeah, I mean, all I can say is, like, before even being a PI, you'll have to start living a PI's life.
Okay, that's great. So I wish you a very wonderful journey ahead with great accolades, and hopefully we will get more, we'll collaborate to move ahead. And
thank you, Yash.
Now I request Dr. Akira Roy to share your slides. So her postdoctoral work is really wonderful. I'm excited to see her postdoctoral work experience and how she is translating her expertise to Indian scientific arena. So with this, please go ahead and share it.
Thank you so much. I'll just share it and see whether it's working. Can you hear me properly? Okay.
Yes, you can make it full screen. Yes,
Okay. So, hello everyone, and I would first start with thanking Sci-ROI for selecting me for this live talks, and also congratulate for the launch of the Sci-ROI at the Red India branch. So as Avijit also said, that I'm quite new in the transitioning, although not as new as him. It's been 4 months. I moved from US, finishing my postdoc to JNCSR Bangalore. Initially, I did my PhD in TIFR Mumbai and in neurodevelopment, and then briefly on translational neuroscience in Seattle Children's Hospital and Research Institute, and then came back here and joined the neuroscience unit as the Ramalingoswamy Reentry Fellow from the DBT. So I will also actually give a brief glimpse.
Excuse me, sorry, perhaps your slide is not moving. Are you on the first slide?
Yes.
Okay, I thought you moved to next slide. Sorry, go ahead.
I think the way I will start is that I'll briefly give you one part of the postdoctoral work that I did, and I would like to move to different avenues with that in my independent laboratory. So different genetic and non-genetic defects actually leads to a lot of neurodevelopmental problems, and mutations in specific signaling pathways give rise to a spectrum of cancers as well as brain and body overgrowth syndromes. My interest during my postdoctoral work was such mutations in a particular critical signaling pathway called the phosphoinositide 3 kinase pathway, or also known as the PI3K-AKT-mTOR pathway, which is very crucial for brain cell growth and development and cell metabolism, cell cycle, etc. And being a neuroscientist, I was more interested in what are the underlying mechanisms of brain overgrowth and associated issues. Interestingly, depending on the extent of mutation, the timing of the mutation, and where the location of the problem is, there's a different terminology is given. For example, like you see here, that brain overgrowth, tiny malformations is called focal cortical dysplasia, while when it comes to like half of the brain, it's called hemimegalencephaly. Megalencephaly means brain overgrowth. And then to dysplastic megalencephaly, which means the overgrowth is in both hemispheres. So this is not just overgrowth, but it is also associated with other severe problems like epilepsy, hydrocephalus, problems with the development of cortical tissue, autism, intellectual disabilities, so on and so forth. And all of these actually have a huge effect on the quality of life of both the patients and the caregivers. And mostly they are non-responsive to current medications, and hence there's a very high demand of non-invasive small molecule therapies to treat these patients. These patients, again, are mostly pediatric. The problem starts in pediatric population, and sometimes it can stay for the lifetime if they are still living. So my interest from the basic neurobiologist point of view is like, I would like to understand the intermediate steps that connects the source of the problem, which is for this instance, genetic mutations, and to the final effects, which is large brain, dysplasia, hydrocephalus, epilepsy. Today, I will talk about hydrocephalus, which is what I would like to continue in my independent lab here. So to understand this, we generated mouse models of the actual human specific pathological mutations in the catalytic subunit of PI3K enzyme, which is called PIK3CA. And very interestingly, and I mean, we are glad that the entire spectrum of human disorders could be recapitulated in our different mouse models. For example, one model shown here, compared to the control littermate, this mouse has a bulging forehead, which is indicative of progressive hydrocephalus. I'll come in a bit what hydrocephalus is, and then expansion of the brain size. Interestingly, we also found that this is embryonically induced. So if the mutation occurs during embryonic development, we see this spectrum. But if this is induced after birth, this spectrum is almost, actually, almost of these are not existing. So now I'm coming to hydrocephalus. Hydrocephalus is nothing but expansion of brain spaces called ventricles, and then accumulation of cerebrospinal fluid in these expanded spaces. And it is actually among one of the most common neurodevelopmental disorders, having almost 1 in every 500 live births. And in India, it's pretty common of the congenital hydrocephalus subtype, almost 12,000 cases per year. And very unfortunately, this only therapy that can still work is invasive and recurrent shunt surgeries that has a very high failure rate. So again, as I said, reiterating that non-invasive small molecule therapeutics is required, and for that, we need to understand what is going on biologically. Additionally, clinically, in addition to these expansion of ventricles, there was also an association of dysplastic cortical folds. So as you mostly know that the human brain has folded brain or gyrencephalic brain. These fold pattern is very unique and is almost stereotypic in humans with some modifications from person to person, but this drastic change is abnormal. And there's a coexistence of cortical folding problems with hydrocephalus was observed both in the human patients as well as in the model counterpart, which was interesting. And so we tried to find this out, that folding as well as hydrocephalus, what is the mechanism of coexistence and can we treat this? So as I said before, that this most of these things are actually embryonic. So we went back, this is postnatal day, what we are seeing here. We went back to embryos and try to understand what are the mechanisms involved. So in addition to proliferation and differentiation defects, that was kind of obvious that increasing folding might be involved in increased proliferation and differentiation. Interestingly, we also found very focal unzippering of the edge, which is connecting or opposing the lateral ventricles, which is shown in the schematic here. So this is the cortex and the hippocampus on top of each other. With cell adhesion molecules, we find that there is a kind of blebbing and also accumulation of molecules at this edge while absent in other places. Interestingly, a molecule called YAP, which is a marker of early ependema that is responsible for the proper connection of this lateral ventricles and the development of cerebrospinal fluid mechanisms, dynamics, I would say, is high and nuclear in structure, while in the control, you can see that it's actually cytoplasmic in structure. So why these problems are happening? We also found that this is not just in the embryos, but this exists even postnatally. So it's not just a developmental delay, but there's a proper pathological issue here, and which is increased as well as the distribution of the molecules went beyond the edge to the tissue of the brain to a large extent. And in addition to that, there's a high increase of progenitor populations, which is also very abnormal in this stage of the development. So bringing back to some perspective of what we were saying, this is an apical cell, and the YAP molecule, which is right now our crucial molecule balancing this act, has a dual role, both as a nuclear form and a cytoplasmic form. The nuclear form helps in cell proliferation and growth, while cytoplasmic form helps in apical cell adhesion. So this balance is critical, and if this gets disrupted, we get the issues what we are seeing. And how is this connected to the PI3K pathway? Well, so it is directly downstream of the mutations where we were having the mutations in. So this is the PI3K enzyme that changes one molecule form to another isoform. I don't want to go into details. And then downstream, the phosphorylated YAP, which is cytoplasmic YAP, changes to nuclear YAP and goes into to cause transcription. So we found that if this balance is disrupted, maybe if we can reduce the nuclear YAP, can the rescue happen? So we used a nuclear YAP antagonist, which is called verteporfin. This is also an interesting repurposing of the drug because verteporfin has been FDA approved and used for reducing abnormal blood vessels in the eye and to treat macular dystrophy. So verteporfin, this is the first study where we use verteporfin in embryos inside the mother. So it crossed the placental barrier as well as the blood-brain barrier. And we could find actually there was a rescue. So this is normal. There was this hydrocephalus, the gap, and then after verteporfin, it got sealed back again. And also there is a reduction, if not the full rescue of the folds. And this is just not morphologically, but even transcriptionally, we could find that the change has happened. Moreover, there was rescue or like a partial rescue of the adherence junction, the cell adhesion molecules we just saw, and change in also the distribution and number of YAP molecules to normal levels. And again, even more interestingly, there was also a reduction of the gyral progenitor number. So more or less, there was almost a 90% attenuation of the abnormal phenotypes with the help of verteporfin. There are more interesting studies that will be coming out, hopefully from the lab, which is just getting set at JNCSR. So what I said, wrapping up, is that so disease regulations, which might be an upstream activating mutations or a YAP knockout, or maybe even other things called like hemorrhage, like injury or infection, that may disrupt this balance, hence leading to ectopic cell proliferation and growth while reducing the apical cell adhesion, and that may lead to ultimately problem with the ependymal development and finally hydrocephalus and death. So what to do? We found one mechanism where the PI3K YAP regulation can stabilize this and get back to the balance. So that helps, and also we don't have to waste our money, time on finding another drug. Obviously, there will be more in the market, but here is one example of that, how a discovery of a new therapy, at least a preclinical therapy, is possible through potential repurposing of already available drugs in the market. So in a nutshell, what I showed here is that mutant mouse model of gain of function or activating catalytic subunit of PI3K enzyme leads to brain overgrowth, developmental hydrocephalus, and other things. The critical period is embryonic, and focal disruption can be altered by nuclear YAP suppression and helping in the stabilization of cell adhesion and progenitor increase. So this part of the work was maximum done in my postdoctoral lab, and I would like to thank my mentor, Kathleen Millen, and the collaborators who helped in both the human and the mouse counterparts of the project. Would also very thankful for the support of the fundings that our lab received. So coming to what I want to do now is that the future research and strategies would be broadly to determine the basic biology and non-invasive therapies to all these spectrum disorders. I just said one of the things today. I also am interested in the evolution of the brain in the sense that there is a dynamicity between unfolded or lissencephalic and folded brain, which is gyrencephalic. As you can see here, the evolution of it is like unfolded, folded, unfolded, folded. So it's not like there is earlier ones or the lower, so-called lower mammals have folded brains and so that is also an interesting way of to identify common mechanisms. And we will be using a lot of different avenues to go into that goal. My broad long-term interest is also in India to bridge basic research with health and pharmaceutical sectors, which is not, as per my understanding, that not that easily available like what we have seen in other developed countries, but that will be very critical to kind of combine bed, like bedside to lab and then back to bedside kind of loop. So I hope that can be possible with the collaboration and platforms like Sci-ROI. I'm also interested to find whether there are convergent mechanisms that are involved in both genetic and non-genetic sources leading to the same thing. That will again help to not to find different medicines for these different problems, but it's actually the same problem caused by different things. It'll be a multifocal way of seeing a problem, and I think that will be quite challenging but exciting. So as I finished my scientific part of the talk, I'll quickly say a little bit about what I went through. I would not say I went through, but at least some of the experiences I faced, which could have been maybe, which made it a little difficult to get a job here. But again, there was some saving grace too. So hiccups along the way. One is that many actually have shared, not only me, that there has been a rare response to application and inquiry emails, which is sometimes for especially non-resident applicants, they get a very stressful, and I'm happy to discuss more if people have questions on that. I, per myself, faced these kind of issues. Like I got into the first application and interview phase, and then they gave a 7-day notice to come and visit and do an in-person interview. Also during a global lockdown, the first wave, they had rigid, like, and these are not small institutes, these are big established institutes of India, and they said we cannot change our policies, we'll think about it, but your interview can wait, but we cannot change it. So I lost these opportunities, and it's again taking time. And then there are something about rolling advertisements where in later on some of the workshops I attended, I came to know that some applications just get removed from the system after a point. Even those can not be even seen. Like they may not be even looked at, but removed because they have just moved some months in the system. But that doesn't mean anything because it's a rolling advertisement. People can send applications anytime. So this is also a mystery. And then COVID happened. So it has been stressful, yet I would say there were quite a few things that happened, and I would really want to call out these. So just before we shut down, as an entire world shut down, the YIM 2020 happened in February, I think, and that gave avenue to what are the new grant processes, applications, talking to people, having mentorship sessions and breakout sessions like this in person was great. And the other thing I would really like to say that Sci-ROI 2020 annual event I attended, and it was a really nicely organized event, one of the possibly the best virtual events I attended that year. And it actually gave me hope that there are people who would really want to solve some of these problems and would like to help each other. And that was a real boost in a little depressing situation I was going through that time without any job things. And then DBT Ramalingam happened, which was really, I mean, I cannot be more thankful to this, because otherwise I wouldn't have come here, because regular positions are now a lot are frozen. And then joining JNCSR, their helpful colleagues is always a bonus. And then getting a little bit of support to get a lab and having a communication, which is like, I am not a regular faculty. I never felt that ever, when I'm always welcome to meetings for my opinions. And I think that helped me to build the confidence that I can possibly survive. So here is the department, and this is my newly made lab. We just moved 2 days ago. It's still, many things are an issue, but we are getting there. So any emails, any collaborations, if you want to have a discussion or want to know the avenues, please email. And there's also PhD and PhD advertisement coming. So please consider neuroscience unit. There's some exciting research happening there. So thank you. I'll close there. Thank you for your attention.
Thank you very much, Dr. Akila. This is so wonderful to hear your research and experiences. And in fact, everyone, we all are sorry to hear that you also missed some of the great opportunities because of the 7 days notice. I've heard kind of stories from several people. That's a really like unfortunate. So from the audience, do you have any question you may ask now?
Achira, I have a question. How did you go about finding a host institute when you applied for some of these fellowships? And was Ramalinga Fellowship the only fellowship that you applied for, or were there others as well?
Okay, so mine is a quite a winding path, but that's one of the most crucial questions, I would say. Host institute is the first thing you need to find before applying anywhere. For me, I had actually started giving informal talks from a quite early phase, maybe from the second year of my postdoc, to have some little bit of networking, continuation of the networking in India after I left, after my PhD. So that time I actually gave an informal talk at JNCSR. They liked me and they said, come back for a proper job talk. So that happened some years later. The job talk with proper chalk talk interactions that happened, and they were interested in taking me as a regular position. I was by then not exactly ready to join, because I was on the verge of finishing a paper and I was committed there. So I said, maybe after some month. That was a naive thing to do, maybe in hindsight. But so we already have a connection there. So later on, I actually didn't intend to apply to Ramalingam at the first go, because I thought I will get a position, but it didn't happen. COVID happened, then freezing of regular positions happened, and so I was in a complete soup. So then this opportunity came, and JNCSR said, do you want to apply? And I said, yes. Can you be my host? And they said yes. Before that, I actually tried Welcome DBT Intermediate Fellowship. I requested the same host. That time they were not sure, so they declined being the host. So it depends. Although they like you, they might not be ready to host you, because hosting means a lot of financial responsibility also. So I would say that it's better to have informal connections and give talks, informal talks, job talks, whatever, and seek in the first go that can you be my host if I apply? I think that's the best way to go, in my opinion. That was my experience.
Okay, thank you very much.
Yeah, so the correct way to do so. Like, I'm also like Ramalinga Swami fellow, so I would just echo what she said. You must have some informal conversation with the host institute professor, so that is
Yes.
very helpful. Okay, any other question if somebody has? So I'll ask if nobody has question. So how did you come up for the proposal of Ramalinga Swami Fellowship? Like, when did you start thinking of it?
So, frankly, there was already this work going on, which I presented today. And I had some ideas that was not getting possible to do because we were also in a financial crunch in US that time for reduction of fetal research, etc. So what I thought was like, well, maybe I will use those ideas in my independent research. So I would say 2-3 years ago, these things were already, I was jotting down points, and then when this opportunity came, I just finalized it.
And so how did you find this transition? So you are in one of the best institute in India. So do you see any difference from like US Institute and JNCSR research culture?
So I'll give you one other thing is like I had been a summer fellow in JNCSR like long back. So that has also changed. JNCSR full setup has changed now. So there is no connection really. But these in new informal talks and formal job talks, I could at least find some of the infrastructures that I would be interested in because my work is completely on mouse models. So not having an animal house was not an option. The comparison between US and here, I would say the bureaucracy is a little, I'm not that diplomatic, sorry, but there is a lot of more paperwork that has to be done, and punctuality is an issue. And also I faced quite a bit of struggle learning the purchase process and the struggles with that. It took time and new things, the new policies coming into place. Even the departments don't know how to deal with it. So these were kind of the additional hiccups while I am in these during these 4 months. Other things is like, I would say the positive thing about here is I think there are way more helpful people. They might be less efficient, but they're definitely more helpful. They're always eager to help. And the experience what I had in US, that I lacked that feeling from where I was. So I would give that to James.
Yes, that is great. So with the time constant, we might have to end and we have to move to next speaker. So before we end, I'll just suggest you one thing. We have a wonderful science researcher here. He's also Ramalinga Swami Fellow. His name is Dr. Somashis Ghosh Dastidar. He's also a neuroscientist. So he might be interested in collaborating with you. So,
okay.
And we are in like in Kasturba Medical College. So you might get a patient sample, wonderful patient sample collection.
Oh, that would be wonderful. I'm actually looking for sources.
So
okay.
We can talk later, and you can connect. I can connect you with him. So you both can go ahead.
Oh, that's wonderful. Thank you so much. Thank you so much.
So with now we move ahead to the next speaker, and she is Dr. Anita Ghosh Dastidar. And actually, this presentation, I'm also involved in this presentation. So this is a collaboration or consortium-based project. So in this consortium, 3 scientists basically involved. One is Dr. Anita Ghosh Dastidar, second is myself, and third is Dr. Somashis Ghosh Dastidar. We 3 came back from US in last 2-3 years, and we were part of Sci-ROI. So we always talked about a theme-based research center in India. So with that theme, we came back to India, and we thought to do something special, what we always talked. And in that light. So thankfully, we got a very good mentor, a professor. With the help of him, we started one consortium-based project, and Dr. Anita is going to talk about that. So she will talk about her research and then the consortium-based projects. So with this, Dr. Anita, please share your slide and share your experiences.
I just want to check once I am audible, right?
Yes, you are.
Okay. And let me know when you can see my screen.
Sure. I can see
Can
your
you
screen. Yeah, you make it full screen.
Yes. And
Yes, please go ahead.
okay. Okay. So first of all, good day and greetings to all of you who are present here. First of all, I would like to thank Sci-ROI for giving me this opportunity to present here in front of everybody. This is a great chance for me to tell what I have been doing in the past and also bring up what I'm currently doing. So thanks for that. Before I start, I would like to thank Dr. Piyush for the nice introduction about our project. So I'm definitely going to talk a lot more about it. But before that, I will briefly talk about what I have been doing in the past. Okay, this is not moving for some reason. Okay, you can see the next slide, right?
Not yet. Yeah, it moved now.
Okay, so so I'm a molecular and cell biologist by profession. I have done my PhD from UT Dallas. I will talk a little bit about my PhD work, what I have done over there. My postdoctoral training has been in UC San Diego, followed by quite a few years of teaching. So I have been teaching in the San Diego Community College District mainly, but then later on I moved on to Durham, North Carolina, where I have taught in the Community College District there, and then finally in NCCU University in Durham, North Carolina. So talking about my PhD work and postdoctoral work, this work that I'm showing here is basically my PhD work, where I have worked on a large-scale genome-wide studies, where what I have been doing is, just a second, I have actually screened more than 4,000 yeast proteins, and ultimately I found out that there are 203 proteins in Saccharomyces cerevisiae that actually alter or change their localization under hypoxic condition, which is a stress condition. What is being shown here in this picture is that different patterns in which you actually can visualize and say which location or which organelle the protein is actually expressing itself. So this was an entire GFP tag library that I had used, and different patterns are shown here. I want to mention specifically that whether it was the aerobic or the hypoxic condition, these are certain examples where no change was found, which means doesn't matter what the condition was, they maintained their localization. So this particular pattern that we see here is nuclear pattern. As compared to that, if you see the mitochondria, it has a very different pattern, as you can see here. And this is being maintained by most of the proteins, whether in aerobic or in hypoxic condition. On the other hand, if we go to, say, for example, vacuolar membrane, it again has a different pattern from which you can say with what location it is. And cytosol is pretty much all over the entire cell, so that's why it looks the brightest of all. Similarly, few other examples of nuclear pore and spindle pore is also given here. What I would like to show here in this figure is that these are those few examples out of the 203 proteins that I talked about, which actually changed localization under hypoxia. So basically, they were synthesized and then they were retained in the cytosol itself. For example, if you see this mitochondrial protein, LYS12, in aerobic condition, this is the typical mitochondrial pattern. However, under the hypoxia, it's just retained in the cytosol, and that's why it looks very different from how it looks in the aerobic condition. However, as soon as we change or bring it back to the normal or aerobic condition, it comes back to its localization, the normal mitochondria. And this thing happens even in the absence of protein synthesis. So when we inhibit with cyclohexamide, even at that situation, they can relocalize back to the normal condition. So these are few glimpses of different proteins, either mitochondrial or ER is shown here. But actually, if you see here, so this image is actually showing the entire 203 proteins. And as you can see here, the majority of these proteins that have changed their localization under hypoxia, they are all nuclear proteins. And they have been categorized according to their functions, depending on whether they are transcription factors or involved in spliceosome, or whether they are RNPs. So different function-wise, they are shown here. This is followed by another major group, which is the mitochondrial proteins. And these proteins are the ones that have changed their localization under hypoxia. But apart from these two major organelles, there are other organelles as well, like vacuole, Golgi, ER, and others, where changes have occurred as well. The interesting fact, and as we will usually assume, is actually the fact that the location change from air to hypoxia is a much slower trajectory. So it varies anywhere from 6 hours to 12 hours. As compared to that, when we bring any protein back from the hypoxic to the aerobic, that is the normal condition, it follows a much faster trajectory, which can occur within 2 hours of time. So with this, I would come to my postdoctoral work, which was in UC San Diego. What I show here in this slide, it really does not do justice to the amount of work that was done here. However, my postdoctoral period was pretty short. I was not part of this entire story, but a very small part of it that I will explain. But basically, this is about this protein called GIV or GERDIN. GIV or GERDIN, it is a GEF protein, which means it has guanine exchange factor. And it helps in the activation of the G protein, so that it converts from the trimeric G protein to the active GI form by separating out of the G beta gamma dimer. This entire process of activation of the G protein can only happen when GIV interacts with the trimeric G protein, but it has a crosstalk with ARF1 GTP as well. So that is what is shown in this entire pictorial. Basically, it begins from the ER ergic region and it ends in the Golgi region. In all this process, GIV plays a very important role. When the GIV is not attached to the membrane, that helps in the vesicular budding, so that the COP1 vesicles are formed, as you can see here. Once this is formed, then GIF can come back and interact again with the COP vesicle, typically beta COP. And after that, it brings in ARF-GAP3, as you can see here. So this is where I had played a role. What I had shown is it actually is the ARF-GAP3 and not ARF-GAP1 that actually interacts with GIF. And that ultimately helps it in activating the G protein in the later stages. So once this interaction happens, then the GIF activation can happen. And after the G protein activation can happen, and only after that the vesicle fusion can take place with the Golgi, maintaining the integrity of the Golgi structure. The other work that I have done during my postdoctoral period is related to a protein called as DEN, domain containing protein 1A, the role of cyclic AMP signaling in its expression. So what is known is under this particular condition of polycystic ovary syndrome, there is a much higher expression of DEN1 than the normal follicles. What I was able to prove is that, so these DEN1, these are actually helping in the process of excessive androgen synthesis, which is found in case of PCOS. So in case of PCOS, what happens is that due to the activation of cyclic AMP signaling, the DEN1 expression increases both at the RNA level and at the protein level. And as a result of this, a lot of other expressions are increased, which are related to the cholesterol and androgen synthesis. So basically, the STAR expression is increased a lot, and that helps the STAR to bring cholesterol into mitochondria, where it can be converted to pregnenolone. From there, it then moves to ER, where it can be converted to androgen. So cyclic AMP activation, and as a result of that further, the DEN1 expression increase brings this entire process into upregulation. With this, I would like to end my past work and move forward to my current work in Mahe, Manipal. My current focus is mitochondria and energy metabolism. My field of work currently are two different fields. So one is ataxia and paraplegia, and the other, of course, is melioidosis that I will focus more upon. Ataxia is nothing but a dysfunction of the nervous system where the coordination of movements is lost. As a result of it, there are effects that you can see in brain and in the neuromuscular junction. And there are a lot of ataxia symptoms that you can actually see by which you can confirm this case. Other case is paraplegia, where the lower limb becomes paralysis. Now, why I'm mentioning these two is that there are two mitochondrial metalloproteases, typically called mitochondrial AAAs, that are involved with mitochondrial quality control, and the deregulation of these proteins can lead to neuronal abnormality, associating it to ataxia, paraplegia, ultimately leading to neurodegenerative diseases. Coming to melioidosis, which is a neglected tropical disease, and I just want to emphasize here that how important or how deadly it is. So yes, it is a very, very life-threatening disease that is caused by a gram-negative bacteria, Burkholderia pseudomyeli. And India shows the highest burden of mortality, which is over 30,000 deaths per year. The national relevance becomes even more with the comorbidity of diabetes mellitus. And along with that, this particular bacteria is actually a tier 1 select agent, and it has this intrinsic multi-drug resistance. Our preliminary studies show that the endemic Indian strains, they are very much distinct and diverse from the regularly studied global strains, which are majorly found in Thailand and Australia. So definitely there is significant difference in the patient demography, in the risk factors, mortality, and morbidity. So this suggests that a detail of work is actually needed. So the need of time is to explore this unique susceptibility of the Indian hosts. However, there is such massive amount of work that can be done that it cannot be done by a single person. So that was the whole idea of why we brought this idea of developing a consortium-based center for studying melioidosis. I would like to start with introduction of my mentor, Professor Chiranjay Mukhopadhyay, who is the Associate Dean of KMC Manipal and Director of Manipal Institute of Virology. He is the main person who incepted Center for Emerging and Tropical Diseases. Later on, as Dr. Piyush has mentioned, that we are 3 researchers all back from US, and we are now thinking of actually collaboratively working on melioidosis, and that is why this whole idea came up. So we already have started talking to different people, setting up collaborations, pan-India as well as globally. So we do have a lot of people interested within KMC Manipal as well. Apart from KMC Manipal, so the entire previously called Manipal University, now called as MAHE. So there are a lot of institutes under MAHE deemed university who are interested in collaborating with us, and their names are shown here. We also have a lot of people from national level who have shown interest, and we are constantly in touch with them. And we do have international collaborations as well from Netherlands, UK, USA, Austria, and Thailand. With this, I would like to very briefly introduce the CETD. So CETD, this was created by Professor Mukhopadhyay and my mentor, founder and executive committee member of the International Melioidosis Network, the expert member of Government of India and Parliament Committee. The main idea was cross-cutting research in emerging and tropical diseases, and melioidosis is one of those. The mission was to act as a center for reference. Dr. Sumashish Dastidar, he focuses mainly in neuromelioidosis, which is a rare but fatal disease. So he is currently focusing on 23 patient-isolated strains that cause neuromelioidosis, and his expertise is on host surveillance evasion and colonization. Apart from that, he is also into neuroscience, so mainly focusing on mTOR biology in aging and age-related neurodegenerative disorders. Our second person in group is Dr. Piyush Bihari Lal, who is here as the moderator here today. So he is trying to understand the host-pathogen interaction, and also there is a lot of work to be done for genome annotation, which he's very much interested in and is trying to improve on that. He also has interest in synthetic biology, so wants to develop better biofuels producing Xymomonas mobilis, and that can metabolize a variety of carbon sugars and also produce eco-friendly byproducts. Myself, I have talked enough about myself, but regarding melioidosis, I'm working at the clinical interface with patient-derived sample studies and analysis. Comorbidity, diabetes mellitus is a great concern, so I'm looking into that as well with respect to altered immune responses. What we all together as an infrastructure and expertise bring to this consortium-based project plans is we annually have 30 to 40 melioidosis cases in our Kasturba hospital. Harvesting bacteria from CSF is easy. MRI, CT scans are always available. We are doing a lot of large-scale metagenomics, metabolomics study. Dr. Sumashish has already developed a mice facility and we do have the expertise of CRISPR-Cas9 and IPSC over here. Dr. Piyush is developing more into the core facility of high throughput sequencing and transposon sequencing. While myself, I'm interested in the fluorescence and live cell imaging, confocal imaging, PAC studies, and energy metabolic studies. I think I'm already running close to the end of my time, but I will try to be very quick regarding some challenges that I have faced after returning to India. I joined MAHI in October of 2019, so that looks like it's quite a bit of time, but time just flies and you never realize. Lab space planning and approval was very easy. Lab space renovation is pretty simple. And all these are because of all the support that I have got from the VC of MAHI, from the dean of my college and associate deans, without whom it would not have been possible. So I'm really thankful for that. What actually has been difficult is turning this into a working lab space. And what I mean by that is, this is what happens. So ours is an academic institution. So unlike the research institutions where a lot of labs are, actually all the labs are doing research and there is a lot of equipment, so sharing is not a problem at all. In academic institutions, sharing equipments and resources is critical, but at the same time very challenging, because first of all, there are not enough resources or equipments, and even if it is there, they are in pockets and places and faraway areas where practically it is not possible to actually continuously move when you are actually doing an experiment. Intramural funding, again, I have been very thankful. We have got great support from MAHI. So they have given me enough funding to set up lab and initiate my lab work. But pandemic, as Dr. Achira said, yes, it has hit us very hard. And it has taken a toll. Things have moved very slowly in the past 2 years. And right after I came, just less than 6 months, the pandemic happened. So, of course, not much could be done. Extramural fundings, yes, I have submitted several extramural grants so far. But what is the real challenge for me is the time constraints. Time constraints to write a good competitive grant, time constraints to generate preliminary data good enough for putting it into a grant with the limited resources that you have. Yes, work-life balance is a big, huge issue along with pandemic. So I have also had my younger son, so he is now exactly 1 year, 3 months old. So I have gone through those maternity-related issues as well. Family-wise, a lot of issues, especially for those families who have kids. I have continuously had nanny issues, unlike in USA where you put them into a daycare and you know they will be taken care of, and they are actually being dealt with by professionals, so you don't have to worry much. Here you get help, but from local people, poor people, so quality-wise that is a compromise which you really don't want to do for your kids. So those kind of things are definitely a challenge. In research, not trying to digress from your main focus area of research is a big challenge, especially with limited resources. So what happens is, if anybody comes to you, approaches you within institute, within university, saying, I have some funds and I want to do this, would you be interested? It's really hard to say no, because you're thinking in terms of publications. But then that ultimately in the long run may not help you. So staying focused under these situations, I felt was a very big challenge. Collaborations, very much important. I have been out of India for more than 14 years before I came back. So I didn't have any connections here, and it's like starting from scratch. So that takes time, and collaboration is even at the students level, at your colleagues level, and at the higher hierarchy. So everything matters, but time you have to really juggle around. Budget, financing, this is in terms of buying equipments, buying consumables. What I found it here is it's a huge process and a very slow process, and there are a lot of consecutive small steps. Not only that, you also have to continuously remind everyone. So this was not what I had an experience in USA. Even as a student, anything, any kit, any chemical that I had to order, I will just do it myself. I never had to ask anyone, and that's what I was used to. But here it's like a hierarchy. You have to talk to separate vendors, ask for at least 3 quotes, send it to purchase. They will call for official techno-commercial meeting with all the 3 companies. So you deal with them, and then purchase will try to negotiate to bring down the prices, and then they will bring up some companies, some vendor who are selling something really cheap, but you know the quality will not be good. So you want to convince them that why you want to do that. So I think I should stop soon. Yeah, this is my last slide. Hello?
Yes, please. Yeah, conclude soon.
Yeah. So all that matters, and for consumables, it's a cyclic process. So every time you have to reorder something, and the entire process starts again, which is a very time-consuming factor. So with this, I would like to conclude, and I would like to thank everyone for your kind attention. Big thanks to all the awesome students who have made my life easier even through all the challenges that we had. Thanks to Mahe and KMC Manipal, thanks to the VC and Dean and Associate Deans, thanks to my collaborators, Dr. Sumashish and Dr. Piyush as well. Thanks to Sci-ROI for giving me the opportunity to talk over here. And thanks to Validate, which is a global network related to emerging and tropical diseases, mainly focusing on vaccine developments, but they sometimes even donate instruments. For example, we won a lottery for confocal microscope, which is like 1.5 crore rupees. So these kind of things can also happen. So thanks to them as well. With this, I would like to conclude. And any questions, if anybody has, I can take them.
Thank you, Dr. Rinda. Is there any question from audience side? So, okay, anyway, we are running late. We are running past our time, but I'll ask one question. So, what is your suggestion if somebody is moving from US to India, especially you are moving to private institutes? So, what's your suggestion for a newcomer? 1 or 2 tips.
So yes, one thing definitely, if you're moving to a private institution, you have to be sure that how much of research they're already into. So as the other speakers have said, so definitely talk with them informally to know what instruments, what facilities they have. In my case, what has happened is that they said that they had many facilities, but when I ultimately came here, it was like one instrument is under one department in a different building, and another instrument is again in another building, and actually you cannot move them to one place. That is the rule. So these kind of things you have to be aware of, and you have to mainly directly talk with them and figure it out ahead of time.
Okay. With that, we are already past time. So if you have any question, feel free to email us, and we are looking for collaborations. If you have different set of expertise, so that will help us. So do contact us, and also any of the speakers will be interested in helping any newcomer or helping in setting up collaborations. So it was wonderful session with listening to 3 speakers. So I thank everyone. And with this, we end today's session, and we'll see all of you tomorrow in next session.
Thank you, Dr. Piyush.
Thank you.
Thank you, Dr. Piyush, and thank you all for your attention.